get to deregulation and bacteria. So actually, we really are going to talk about bacteria again, but for this chapter, we're finished. So in your notes, that's for the bacterial genetic stuff we're covering right now is done. But I wanted to give you guys a couple of examples to have in your notes as you work through homework problems and thinking about the exam and that sort of stuff. So one of them is a transduction problem. Okay, so what does this make you think of right away? How are we transferring DNA back and forth between bacteria? With a virus, right? We're using phages, so this should make you think of the viruses moving back and forth. You can instantly think about the biology behind it so you can understand the genetics. I'll let you know the information that's given on this problem. So you're given a donor genotype and a recipient genotype. Your donor is plus for pure and NAD and minus for PDX. Recipient is the opposite genotype. You're told that the donor allele pure plus is the one that's initially selected for. So this would be after transduction has occurred. Okay, so you mix these two, you mix a donor, you mix a recipient, you add the phage viruses, you add the phage viruses to your donor, let them infect, and then you add that mix, the phages in particular, to the recipient. Okay, so after transductions occur, you then put them on plates that are selecting for pure plus. Those recipients who've received pure plus from the donor, okay? And then a new additional 50 total transductants are selected for the other allele. So you're given a table of this data. So you have all these 50 transductants. So in the left-hand column, you have genotypes, and then on the right, you're given numbers of colonies. Because often, again, in bacterial genetics, we tend to not think of them as individuals, we think of them as colonies. Okay? We count them as colonies as opposed to numbers of cells. But one could argue that you can kind of multiply to it should be the same ratio, essentially, right? If you have numbers of colonies, each colony hopefully represents a similar number of cells. Okay. So we have the following genotypes in our 50 offspring, essentially, that we're counting, or 50 recipients. So the first is NAD plus, PDX plus, and there's 3 colonies. Second is NAD plus, PDX minus, you have 10 colonies for that. Then you have NAD minus and PDX plus, and there are 24 colonies. Okay. And then NAD minus and PDX minus. And you get 13 colonies. so that's the information you're given. Okay, you're told the donor genotype, the recipient genotype, you're told that you're initially selected for pure plus, and then these other 50 are for the other allele selections. Okay. Part A is asking for the co-transduction frequency. Of the pure gene and the NAD gene. So automatically, if you're asked the frequency of anything, what are you going to do? What numbers are you going to work with? How do you get frequencies? Percent. So we're going to take some number and divide it by what?
Total.
A total. So these are ways to kind of train yourself. I know maybe it sounds kind of silly, but these are ways to train yourself to be fast at recognizing these problems. If you see questions asking for blah, 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 frequency, in your head, you know you're going to have to take some kind of number and divide it by some total number of things, okay? Whether that's progeny, colony, bacterial colonies, or whatever. So we instantly know we need a frequency, so we're going to have some number divided by 50, our total progeny number, okay? And what we really want to know is, okay, what's this number? We're looking for pure and NAD, and we're looking for co-transduction frequencies. Okay? So that means your donor, whatever it's giving, so in this case we're focused on pure and NAD, how often these two genes get picked up by the phage and then transferred into a recipient bacterial cell. That's the co-transduction frequency, right? So we want to know how many times these two alleles are inserted and stuffed into the phage virus head, and then it moves along and infects the new recipient cell and shoves those genes, those alleles, into the recipient cell. Because the recipient's always going to be this genotype, minus for pure, minus for NAD. So in this case, we're looking for pure plus cells and that are also NAD plus. Okay. How do we know which of these are pure plus?
All of them.
All of them, yes. I'm glad you're right. So like when you're told, so sometimes you might be told in more of a sentence structured format versus in the table, but you're told right here the donor allele pure plus is initially selected for. Then the other 50 transducted. So really if you want to write in your notes, if you were given a problem like this on the exam, in parentheses, right, we could write pure plus for every one of these genotypes. They're all pure plus. And that's something I like myself personally, if I was on an exam, I would literally write, because I tend to get confused and mixed up by these different alleles. So I'll write in there on the table, pure plus, pure plus, pure plus. So I have 3 genes, 3 alleles for each of the genes on there. So I don't get confused or mixed up by moving too quickly. I'm very methodical about this.
So you're looking for the pure plus and the NAD plus?
Yeah.
So we can.
So we could count that first one, right? 3. What else? 3 and 10. Okay. Both have pure plus and NAD plus. They both had the phage virus picked up those 2 alleles from the donor and put them into the recipient. So your final answer is 26. That's your co-transduction frequency for pure and NAD. Does that make sense, everybody? I know I rushed the last part. I kind of just, we looked at the table and rushed through it. Okay. Second question. So same thing. Solve for your co-transduction frequency. But this time, instead of pure and NAD, you're looking at pure and PDX. So again, you're going to look for all the places that you have the donor allele for pure and the donor allele for PDX. And so this is where you need to be comfortable with not always thinking about your donor being all plus plus plus, recipient being all minus 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 or whatever. They can have mixture of alleles, right? Pluses or minuses. So in this case, your donor is actually minus for this allele for this gene. Okay. So in this case, you're looking for places where they're pure plus, which is all of these, and also PDX minus. So if this were a nutrient, you're actually making your recipients now be auxotrophs instead of prototrophs, or I'm sorry, yes, you're making them be, they were for PDX if they were plus, then they could grow without you giving them that nutrient. But now when you introduce this donor DNA, you're making them no longer be able to grow without you giving them that nutrient, right? Well, what numbers on here, where do we see co-transduction of pure and PDX? Yeah, so 13 and 10, right? PDX minus, they're all pure plus. So you, again, add up 13 plus 10 and divide it by 15. And you get 46%.
Question.
Yeah.
Just under the impression from the last lecture that after the donor gives to the recipient keeps what it can already do and then takes on what it can. I was under the impression, but it's no matter what the donor is, that's what the recipient's going to be. It doesn't keep its ability for PDX plus.
I see what you're saying. It depends on whether there is recombination between the strand, the amount of the DNA that's being introduced by the virus. So in this case, yeah, to be most correct, you would have to consider it having recombined that whole strand, so that if, like if these were two different strands of DNA, your recombination occurred on the outsides of it. So now it's exchanged. It didn't keep its original genotype. Yeah.
There is situations though where it does keep its abilities?
For the most part, you guys can pretty much assume that it's exchanging it.
Completely.
And if it's ever unclear on exam, come ask me and say, am I to assume that this is not, but I usually try to anticipate that, put it in the wording. But in this case, you're, exchanging.
Okay, completely.
Yeah. I have a part C, but it's like way down the bottom of the board. Any other questions about part A and B? Yeah.
Why are we, and this might kind of sound stupid, I'm not understanding it, only looking at the donor, like, genotype, because we're taking the per plus and then the per, PDX minus. So why are we selecting, why are we focusing on the recipient is what?
Because the recipient is going to receive, maybe it's the naming system donor and recipient that's throwing you off, but the recipient is called that because it's actually going to receive all those alleles from the donor. So kind of that's getting a little bit at Dad's question. So if these are two cells, all of these alleles here are actually getting transferred into the recipient. The way we've written it up here is before transductions occur. So this is the recipient, how it looks before it gets the new DNA introduced. Does that help a little bit? Not a dumb question, that's a good question. That's not a dumb question, that's a good question. Yeah. Any other? Okay, my last question was just, well, hard to write up here, but essentially, which of these loci that you just calculated co-transduction frequencies for? So you don't have to draw a full map, but just answer which one is closest to the pure gene. I'm asking you guys, which one's closest? PDX. So you guys settled on PDX and why?
Higher percent.
Higher frequency of co-transduction. And what did we talk about last time? The closer together two genes are, the more likely they're both stuffed into that viral phage head together. Right? Because if they're really far apart, that means there's a much greater distance, length of DNA that needs to be all bundled up together and stuffed into the viral head. So they're going to co-transduce more frequently. You're going to see a higher percent of co-transduction on two genes that are close together, that are actually located on the DNA close together. So PDX is the closest to pure in this example.
Is that the opposite with like the chromosome? Because the further they are, the more likely they are to.
For recombination? Yeah, that what you just said is correct. Yeah. Yeah. Closer together the two genes are, the more likely we'll see co-transduction, and less likely you'll see recombination between them. Yeah. And that part's true for genetic or bacterial genetics as well. Okay. While I'm erasing, any other questions about, that's the end of this problem that I have for you guys. Just to give you an example of the kind of stuff you might do with co-transduction type of problem.
Yeah. That would be nice to know.
Alright, so my other problem I made up for you guys has to do with complementation in bacteria. Okay, and specifically it has to do with the biosynthesis of arginine, but you guys don't really need to know much about that. But the biology behind it is that you're looking at a pathway, okay? How this molecule is actually synthesized, and it turns out it's in a pathway. It takes a bunch of enzymes to make this happen. Okay, and maybe you're a researcher, and you want to create mutants in these enzymes. So if we just are comfortable with the fact that you don't need to know this full biosynthesis pathway, but that it does take several enzymes, okay, are involved in this process. And you're trying to generate mutants in each of these enzymes. So what you do is you take a bunch of your bacterial colonies and you metagenize them somehow. Okay, so you throw UV light at them or whatever, some kind of chemical. That part you don't have to worry about quite yet. We are going to get to, in this course, we'll talk a little bit about specific types of mutations and how the cells can repair those. So that's coming up. But somehow you're going to metagenize the cells. And then you're looking for colonies that can't grow unless you give them arginine. Okay, so essentially, these guys are all auxotropes for arginine. And then you take those mutants, so you look for those mutants, that would be, essentially when you metagenize and look for colonies that can't grow unless you provide arginine, is that a selection or a screen? Yeah, you're doing more of a screen, but you're selecting if you don't provide arginine. So if you try to put them on plates that are minus arginine, that's a form of selection. And then you're kind of screening as well. So that you could argue, I think you could argue that both of those terms would apply to this last statement. So you do this, okay, and then, yeah.
I'm confused. So, but you're not putting them on plates that's
You're putting them with minimal media, essentially.
minimal media.
No arginine right now.
Yeah, no arginine.
But they can't grow unless they have arginine. Maybe.
So none are gonna grow right now.
Yeah, none would grow if
None would
you,
grow.
if you did not, if you selected for them by not providing arginine, then none would grow. And then if you replica plated it and added arginine in, and all of a sudden you see growth.
Oh, so you're comparing different plates.
Yeah.
Okay.
Oh. So after you find these, so you use the process that we talked about last time with the replica plating and selection. Then you find some of those colonies that can't grow unless you provide them to arginine, and you cross those mutants. Okay? Because really we're talking about complementation, right? So we need to look to see whether, what's the premise behind complementation? What does that mean? What are we looking for? Yeah, whether your mutant or mutation is on the same or different genes. So you're crossing mutants and looking for what in the phenotype. Okay, you're looking for normal, you're looking for wild type. In this case, you're looking for those that can grow without arginine. Okay, so you cross a bunch of mutants. Okay, those that you found through your selection and your screening. You're given a growth chart. So this is a really common way to display bacterial results. So essentially, whenever you're crossing a bunch of mutants, this is a common way to depict this, is you would have a wild type strain as your control, and you would cross that, of course, to another wild type strain. Okay? And then you also have all your mutants. So in this case, the mutants are just numbered arbitrarily 1, 2, 3, and 4. So this table, you're showing the crosses of each mutant, one going horizontally, one going vertically. So you cross wild type to wild type, you cross wild type to mutant 1, mutant 2, mutant 3, mutant 4, etc. Okay? And you're looking at whether there's growth or no growth. So this is indicated by a plus or a minus. So plus means growth, and minus means no growth. So if we cross a wild type to a wild type, do we expect a plus or a minus? If it's just wild type normal, we expect it to grow, right? So we expect a plus. Maybe that should just be normal, can grow regularly on minimal media. And then the rest of the data, and then you're also going to, so you're going to do pairwise comparisons for each one of the mutants. So you're crossing wild type as your control. This is essentially your control line, right?
So everything bred with the wild type will
Yeah,
be wild type.
presumably everything bred with your wild type, you should always see, that should be your, if you were really running this in the lab, you would expect to see growth and all that, and the data supports that here. Okay, and then we're going to cross each one of these to itself and to the other mutants. So again, looking down, every time you cross a wild type to a mutant, we should get growth. So this is data that's actually going to be provided to you. I'm just kind of walking through the table slowly so we understand how to read it, how to look at the data. Where else would we always expect to see pluses?
No restrictions.
And
So
they
what-
go against themselves.
Yeah, so when you cross the same mutant to itself, right? Because what was our definition of complementation? How do we, if they're in the same gene, if you have a mutant that's in the same gene, right? just said. So if you have a mutant in the same gene, you cross it to itself, is
Then
that
they'll stay
complement or
mutant.
not complement?
No.
Not complement, right? So whenever you're same gene versus different gene, right? This is from your notes from last time. If you cross two mutants and they're in a different gene, then we get that. We're looking for wild type phenotype to come back, a normal growth pattern to come back. That's the two mutants are complementing each other by providing intact genes in all the areas. So if they're in different genes, you end up with a wild type phenotype if you cross two mutants. Right, so this is from your notes. You have this in your notes from Wednesday. So actually opposite. So when you cross them, always whenever we cross them to each other, they're in the same exact gene, same exact mutant, so we'll never see normal wild type growth from them. Okay, so you always, when you cross them to each other, 1 to 1, 2 to 2, 3 to 3, and 4 to 4, you have a no growth phenotype. Because you're crossing two mutants that have a non-functional mutation in the same exact gene. So whichever enzyme that is of arginine biosynthesis, it's stopping synthesis of arginine. These can't grow unless you provide arginine. Okay. Does that piece of the chart make sense?
Yes.
Okay, so the rest of it, you'll just have to be given this information, and I'm going to give it to you here. Okay, so you're given the following pluses and minuses in the data. And then how you organize this data is you group them all up into what's called complementation groups, okay? So you're counting up those that do not complement. So it's a little bit backwards because you're adding complementation groups. This term means what you're doing below here is adding those mutants, those crosses, okay, that do not complement. Okay, so therefore, what about the gene? Are they in the same or different gene if they don't complement?
Same.
Yeah, so essentially you're saying therefore same gene. Same enzyme in this arginine biosynthesis pathway. So where do we see that happen? What
1 and 2.
Yeah, so we see 1 and 2,
And 3 and 4.
right? Because here, if we look at wherever we cross 1 to 2,
And 2.
we get a minus, we get no growth. They do not complement each other. You still have a mutant phenotype. They're still not able to grow unless you provide arginine. And then 3 and 4, we see the same thing, right? So 3 to 3, we expect that same mutant, same gene. 3 cross to 4, and same thing with 3 cross to 4. We get no growth. They do not complement. Therefore, they are in the same gene. And that's, again, another tip on approaching these problems. This is the kind of stuff that if this was written in like a sentence format, it says a minus equals no growth and a plus equals growth. Like, I'm the kind of person, I literally would write this right next to my chart. Plus means growth, minus means no growth. Or whatever works for you. Minus means they do not complement. Write that right in your own handwriting, right next to the chart, and make sure that you can come back to that. And maybe that sounds silly, but I think when we try to take shortcuts is sometimes where we have errors on our exam. Some of you guys are just freaking rock stars, and you guys can like do all this stuff in your head, but the rest of us slowly, mortal individuals. I write notes like this to myself. I do that. Don't feel like that's not something not a good option for you. And I stacked them this way on purpose. So I put them kind of in a line, 1, 2, 3, 4, because what does this tell you about how many numbers of genes are represented in our mutant groups here?
Two.
Numbers of genes. So when we add complement groups, those mean they're in the same gene. Yeah, so right, we have one complement group here, and a second complement group here. So this is really saying that you have 2 genes basically represented by your group of mutants that you made. So basically, even though you number mutant 1 and 2 as separate numbers, you're calling it mutant 1 and mutant 2, those are the same mutant. Okay, for the purposes of running through this exam, or running through this problem, it might be different mutations, okay, because we just randomly generated mutants by mutagenizing them. But whatever it is, it's creating that same gene to be completely non-functional. So even though we separated them out as mutant 1 and mutant 2, they're basically the same type, phenotypically the same type of mutant. The mutation has happened in the same exact gene, the same enzyme. So 2 genes represented, you made successfully generated mutants for 2 of the enzymes in this pathway. Okay? That's kind of the conceptual thinking, your final answer for this. If you're thinking about what does that mean for this biosynthesis pathway, that's what you did. Based on looking at whether those genes complement and give you back a wild type phenotype or not.